with me. Hope you can hear me. Before I get started, I just want to talk to you guys about something that I know is dear to every Canadian in this room, I think, and that is maple syrup. Now, I had the chance of spending some time in Ontario during my time in high school, where I learned about the intimate relationship you guys, or some of you guys might have with maple syrup. And I also understood that some of the people in Canada are always kind of worried about the growing demand in maple syrup that is happening. And so I thought this prompted a research question. And I did my research, and I'm very happy to inform you that there's you don't have to worry. The maple syrup production is keeping pace with your consumption. You see maple syrup production in Canada over the last few years in million gallons. But of course, this prompts the question, what is behind this excessive use of maple syrup? And so if you know me or worked here long enough, you know that my family has a very close relationship with Canada. Both my sister and I spent some time in high school in Ontario. Both my parents and my cousin Stefan, whose son Jan is here with us today, all the way from Germany, have spent some time in Canada and worked here with some of you guys. So of course, this raises the question if maybe it's my family that's behind this maple syrup consumption. And I have an answer for you. Indeed, it seems that that is exactly what's happening. Sorry, I have to go back. That the days my family has spent in Canada seems to correlate pretty well with the maple syrup demand here in Canada. I just want to point out that little spike at the end in 2024. That is when my wife will be arriving here in Canada to join me. So I do hope that the maple syrup production will keep up with this. I really hope, because we're not going to go easy on it. So enough about my family. Today, I want to talk to you guys about DNA methylation profiling in gynecological cancer research and diagnostics. And I want to start by giving you an introduction to DNA methylation-based, array-based DNA methylation analysis and what's behind the concept of using that technique for tumor classification and profiling. In the second part of my presentation, I want to talk to you about how we can use this technique actually in diagnostics, and an example of endometrial stromal sarcoma, a uterine sarcoma entity. And then lastly, I want to talk to you about how this methodology has helped us greatly in informing biology and lysosomal-associated neoplasms, the main research topic I'm working on here with David and Yemen and many of you guys. So let's get started. To get started, I thought I'd give you some insights into pathology. I'm a pathologist by training. This is a case as we would, encounter in daily practice. This is a biopsy from the uterus of a 64-year-old female with postmenopausal bleeding, and it was clinically found to have a uterine mass. And you can appreciate that on this slide, there's a solid mass. Not much we can really tell from this magnification, but if we go closer, we can see that this tumor consists of sheets of large cells that don't really give us any hint at what they might be. Usually in pathology, we look for arrangement of cells that look like a carcinoma or like a sarcoma, and that way we figure out what type of tumor this is. This tumor really is not telling us anything about it. And usually then we can use immunohistochemistry, so expression of specific proteins that might give us a hint at a lineage differentiation of this cancer. But also this cancer is not really telling us anything. It's not expressing anything. So what we're left with is an undifferentiated cancer of the uterus. Now, thanks to many of you guys here in Vancouver, we understood that this undifferentiated morphology, not only in the gynecological tract, but anywhere in the body, is often associated with a deficiency in the SWI/SNF complex. And the SWI/SNF complex is a multi-protein complex that's involved in chromatin regulation, so guiding RNA or gene expression in cells, and disruption of this complex by inactivating of one of these core complexes, core proteins, will lead to aberrant gene expression profiles. And indeed, on the right-hand side, you can see the cancer we just looked at is deficient for SMARCA4, so shows core SWI/SNF deficiency. You can appreciate a vessel where the endothelial cells still express SMARCA4, the tumor is completely negative. So we're dealing with a SMARCA4 deficient uterine neoplasm. And I think, that's great. Is that helping us much? Unfortunately not. In the gynecological tract, there's multiple tumor entities that can show exactly this morphology and this loss of SWI/SNF proteins. I've given you 3 examples here. It's undifferentiated endometrial carcinoma, SMARCA4 deficient uterine sarcoma, small cell carcinoma of the ovary, hyperglycemic type. All of these are SWI/SNF deficient. And if we look at the morphology, we can see, you know, even an expert, probably like Blake, who's in the room with us here today, wouldn't be able to tell these 3 cases apart, I would guess, just by looking at histology. And so in pathology, as I said, we typically use immunohistochemistry. This is an example from a study we published in Journal of Pathology 2022. You can appreciate that it doesn't really matter which tumor, all of these tumors can show loss of SMARCA4. So it's not really a good marker to differentiate these cancers. What about clinical information? That might be helpful. Indeed, it is. Typically, undifferentiated endometrial carcinoma appears or is found in older patients. However, there's somewhat of an overlap. So if you have a patient that's 50 years of age, there's not really any good evidence that can help us to figure out which tumor type this is. So if we look at this, of course, the question arises, is it then important to really differentiate these tumors? Or maybe they're the same thing if they have the same molecular alteration, similar clinics, they look exactly the same. Why do we even need to distinguish this? So over the last couple of years, you know, a major theme in cancer research has been that the oncogenic potential of a cancer cell is mostly driven by alterations in genes, such as tumor suppressors or oncogenes, and that was one of the very key features that was driving the potential of a cancer. But in recent years, we understood that there's more to this, and there's other factors influencing the oncogenic potential of a cancer cell, such as the tumor microenvironment. So stem cells in the tumor microenvironment, interactions between cells, autocrine, paracrine, inflammatory cells, and thirdly, also the cell of origin. So cell of origin can dictate which alterations are needed for a cell to move from a non-neoplastic cell into a cancer cell. And so also here in Vancouver, we used lots of techniques such as sequencing techniques over the past few years to study the genome, alterations in cancer cells, tumor microenvironment. We're now using spatial analytic tools to understand the microenvironment, but not much studies have been done on cell of origin in cancers and differences between that. Usually in histology or pathology, we've used the microscope to look at the cancer and infer what type of cell this tumor might have developed from, like an ovarian carcinoma from an ovarian epithelium. But I want to present to you today DNA methylation as a great tool that is able to study differences based on a cell of origin in cancers. So what is DNA methylation? DNA methylation is one of the simplest epigenetic marks in the human genome, by which a methyl group is added to CpG dinucleotides across the genome. And the distribution of these marks can have an influence on gene expression. A very famous example is MLH1 promoter methylation, which some of you might know from endometrial carcinoma, whereby the position of these methylation marks in the promoter region of that gene confers a gene silencing. So these methylation marks are deposited all over the genome, and this is pretty much a dynamic process. This graph shows you the methylation levels at embryogenesis, so in developmental stages during embryogenesis, and you can appreciate that during embryogenesis, many different proteins are involved in a de novo methylation of the human genome. So these different methylation marks are de novo distributed all across the genome. The distribution of these marks contributes to cell differentiation, organogenesis, and somatic tissue differentiation. So we can appreciate that different cell types in our body are defined by very distinct, very specific DNA methylation patterns. And we can appreciate this perfectly in this graph published in Nature last year by Läufer et al. This is a circle plot, an unsupervised clustering of DNA methylation data. And you can appreciate, it's a little bit small for you to read, I think, but you can appreciate that different somatic tissue types, cell types of the human body cluster based on their distinct DNA methylation pattern. And understanding that was a big step for what I'm going to talk about today. So it might seem counterintuitive, but these DNA methylation marks are extremely stable alterations. And because that's the case, they are retained through tumorigenesis. And that means that even in a cancer cell that has gone through tumorigenesis, these DNA methylation patterns of a cell of origin are retained. And we can actually use those patterns also in cancer to classify tumors. And this is a very important study on the right by Kappa et al. Published in Nature 2018, where they used DNA methylation patterns of different tumors to classify central nervous system tumors. So 2D clustering, a UMAP, whereby every dot in this plot resembles a tumor sample. And you can see, based on clustering analysis of DNA methylation analysis, you see that tumor types, distinct tumor types, cluster together in a grouping fashion. So how do we analyze DNA methylation data? Seems kind of complicated. What I have used in the last years for my research is the Illumina EPIC array, which offers an easy and inexpensive pipeline for quote unquote whole genome DNA methylation analysis, whereby bisulfite treated DNA is loaded onto this chip. And after allele specific primer annealing and single base extension, this chip is read out in an optical reader, which results in a methylation level of 850,000 sites across the whole genome. So we'll receive a call of 0, non-methylated, 1, this site is fully methylated. And using this data, we can perform different analyses. Most of what I'm going to talk about is heat map analysis, clustering analysis, whereby we use multiple, let's say 10,000 of these sites to infer differences between tumor groups, similar to what you might have already seen using RNA expression profiling and clustering analysis. So can we use this for the case we just looked at this undifferentiated SWI/SNF deficient carcinoma? We still don't know. Is it an undifferentiated endometrial carcinoma? Is it a SMARCA4 deficient uterine sarcoma? Or is it an ovarian neoplasm? And that is exactly the question that we asked in a study in 2022. Can we infer different cell types of origins for these tumors using DNA methylation analysis? And to our surprise, this worked fantastically. DNA methylation profiling can, in fact, distinguish between these different SWI/SNF inactivated cancers. You can appreciate on the left, again, a circle plot, a clustering analysis, and in the right-hand side, the two-dimensional representation of that clustering, whereby DNA methylation clusters correlate to these distinct tumor types and not the individual SWI/SNF inactivation site. So what does this mean for our case? This is what we had a look at already. It was a 64-year-old patient with a uterine mass, undifferentiated histology and genetics. We knew it was SMARCA4 deficient, microsatellite stable. So this kind of, if you know what you're talking about, you would think this patient is too old to have a SMARCA4 deficient uterine sarcoma. But in fact, by DNA methylation analysis, this was a pretty clear case that was showing a DNA methylation profile along other SMARCA4 deficient uterine sarcomas. Okay, so much for my the first part I wanted to talk to you about. For the second part, I'd like to give you a hands-on example on how we can use this technique in clinical practice. So most or much of my research is focused on uterine sarcomas, so malignant soft tissue tumors of the uterus. And back in the day, we usually classified uterine sarcomas based on how they looked. We differentiated tumors that are derived from smooth muscle and tumors that were derived from endometrial stromal. So we thought today a good way to classify these tumors is actually one, intergenomically complex sarcomas. These are typically leiomyosarcomas that have TP53 mutation, RB1 mutation, show lots of chromosomal aberrations. And on the other side, intergenomically simple sarcomas. And these genetically simple sarcomas are a big field in research. There's been many new alterations that have been identified over the past few years, and we think define very distinct tumor entities. And for my example, I want to focus on endometrial stromal sarcoma. Endometrial stromal sarcoma is the second most common uterine sarcoma type, which we subdivide into subtypes of low-grade endometrial stromal sarcoma So uterine sarcoma entity that shows low malignant potential. This patient usually get diagnosed. It may recur 10 or even 20 years later. And on the other hand, high-grade endometrial stromal sarcoma that is typically diagnosed at a higher stage and may progress clinically. And so the major progress we have made here is, rather than relying only on morphology, we now know that these groups are defined by recurrent genomic alterations, mostly gene fusions. So these are the primary alterations that we can use diagnostically to infer the class of endometrial stromal sarcoma. And these tumors may also show secondary alterations. In the case of high-grade endometrial stromal sarcoma, amplifications of oncogenes and deletions in tumor suppressors. We've shown last year that a CDKN2A lesion in these tumors confers a poor outcome. However, there is some caveats to this. Unfortunately, some of these tumors can show an unclear histotype. So low-grade endometrial stromal sarcomas can show features that make us believe they might resemble high-grade endometrial stromal sarcomas. High-grade endometrial stromal sarcomas can show low-grade morphology. And to complicate things, there's a few cases that have been reported to harbor gene fusions that harbor genes that are usually rearranged in both groups. So it's not really clear what we should do with these cases, how we should deal with this. And so in another study from 2022, we actually studied this conundrum using DNA methylation data, and we were very surprised to see that these two tumor types show very, very distinct DNA methylation profiles, leading us to believe that they are actually distinct tumor entities and not part of the spectrum. It's not a low-grade endometrial stromal sarcoma that at some point moves into a high-grade endometrial stromal sarcoma, but these are biological distinct entities. And furthermore, these unclear cases with unclear histomorphology or unclear gene fusions, we were also clearly able to classify into these groups. So it seems that DNA methylation-based profiling in these tumors seems to be a very good discriminator between these two groups. And so I think, it would be great if we could use this methodology for diagnostic purposes. And I'm very happy that in 2021, I was able to contribute to this study led by Christian Kölsche and Andreas von Daimling from Heidelberg, where they created a DNA methylation classifier for soft tissue tumors, which also included low-grade and high-grade endometrial stromal sarcomas. And from this data set, they built a classification tool whereby you can upload one of this epic array data sets into an online classification tool. The tool compares your data to these reference cohorts and will give you a classifier score. So this is an example. I used a high-grade endometrial stromal sarcoma, analyzed it by epic array, uploaded it, and as you can see, the classification tool will give you a match score. In this case, showed a 0.99% match with the reference cohort of high-grade endometrial stromal sarcoma. So this is still a research tool at this point, but it's it might turn into something we can actually use as a diagnostic tool in clinical practice. So for the third talk, for the third part of my talk, I want to switch gears and talk about embryonal rhabdomyosarcoma. Embryonal rhabdomyosarcoma is a uterine or is a tumor entity that is arising all over the body, typically in very young patients, children, young adults, but it also can arise in the uterus. And this often affects older patients, as saying in middle-aged women rather than children. And an interesting observation of the past few years has been that these embryonal rhabdomyosarcomas, in contrast to embryonal rhabdomyosarcomas of other locations, almost exclusively harbor DICER1 alteration. DICER1 is a gene involved in microRNA biogenesis that is disrupted in these cancers. And so we were very much interested to see what was behind this. Why is DICER1 only altered in uterine embryonal rhabdomyosarcomas, but not at any other location in the body? And of course, again, we turned to DNA methylation, and we were again very surprised to see that DICER1 mutated embryonal rhabdomyosarcomas seem to be defined by a very distinct DNA methylation signal. On the right-hand side, you can see that cluster, the orange cluster down below, DICER1 mutated embryonal rhabdomyosarcomas, above other types of embryonal rhabdomyosarcomas, including non-DICER embryonal rhabdomyosarcomas in yellow, alveolar rhabdomyosarcomas in purple. These are typically fusion-driven, and MyoD1 mutant rhabdomyosarcomas. So this was very, very surprising, and we thought about this for a while. And then, of course, you can ask this question: does this mean these embryonal rhabdomyosarcomas of the uterus may be defined by distinct cellular origins? Yes, that's possible. That's what the data shows. But is it really interesting to anyone? And we were thinking about this, but we thought about it for a while. And this is, I think, where the story gets really, really interesting. Uterine embryonal rhabdomyosarcoma with DICER1 mutations are associated with the so-called DICER1 syndrome, which is associated with up to 30 different neoplastic lesions. It's a tumor predisposition syndrome. And on the left-hand side, you can see two alleles of the DICER1 gene. Typically, we have two functioning alleles of this gene in the DICER1 syndrome, and this was identified by Hill et al. In 2009 and published in Science. In kindreds that have a predisposition, a so-called DICER syndrome, they show an inactivation in the germline of one of the DICER alleles. So this is typically a frame shift or nonsense mutation that is sitting in the germline. Now, what was very confusing at the time is that in other tumor predisposition syndromes, such as BRCA or Lynch syndrome, typically a lesion development would be associated with the inactivation of the second allele. So both alleles are inactivated. But for some reason, that was not the case for DICER1 syndrome. This was a massive conundrum until in 2012, David's group here, and some of you people were probably involved, identified somatic missense variants in hotspots of DICER1. So showing that in this case, one inactivating mutation in the germline and a somatic missense variant in the other allele are associated with lesion development. Now, what type of cancers do patients get if they have DICER1 syndrome? There's many, many, many lesions, up to 30. These typically affect younger patients, children, and young adults. And what is interesting again here is that if you look at other tumor predisposition syndromes, such as BRCA or Lynch syndrome, tumors people get like colorectal cancer, breast cancer, ovarian cancer. These are cancers that are also frequently observed in patients that are not part of a syndrome, so as a sporadic cancer. Whereas in DICER1 syndrome, many of these cancers are very, very highly or tightly associated with the syndrome, so almost exclusively occur in patients that do harbor this germline inactivating mutation. And where it gets even more interesting is that if we look at these different cancers that can arise in the brain, in the head and neck area, in the lungs, in the kidney, in the uterus, has been known for quite some time that they do share histomorphological features. So if we look at them through the microscope, they look eerily similar. It's almost at some point, it's impossible to tell them apart. Just to give you an example here on the next slide, these are three cancers that arise in patients with the DICER1 syndrome: the pleuropulmonary blastoma, a lung neoplasm, most frequent malignant tumor in childhood; anaplastic sarcoma of the kidney, obviously a kidney neoplasm; and embryonal rhabdomyosarcoma, as we have just looked at in the uterus. If we look at the morphological features of these cancers, we can appreciate that one, they all compose a mesenchymal component, almost looking like a sarcoma. These are spindle cells, solid cells that show atypical features such as hyperchromatic nuclei that look atypical. Where this mesenchymal component meets an epithelium that is present in these organs, where it develops, it shows a so-called cambium layer, so a condensation of these malignant cells beneath the epithelium. And then in some of these cancers, we can also see rhabdomyoblast-like features. So some of these tumor cells will start looking like skeletal muscle. They will become big, showing a strongly eosinophilic stroma. And some of these tumors also show a chondroid features, a differentiation. It looks like fetal type cartilage, as we would see in a developing body. So that is very, very peculiar, and many people have thought about this problem. about this issue. And when we identified these distinct DNA methylation profiles in embryonal rhabdomyosarcoma, we thought, isn't it possible that maybe all of these neoplasms actually share a same cellular origin? And to test this hypothesis, we performed a large study with many people from around the world, including Canada, which was published last year in Nature Communications. And in this study, we studied over 530 neoplasms by DNA methylation analysis. Many of these neoplasms were tightly associated with the DICER1 syndrome, and by sequencing were also shown to harbor DICER1 alterations. I just want to go through this plot with you. This is, again, an unsupervised clustering of DNA methylation analysis of those 434 tumor types. And what you can appreciate is that many of these tumor types, which are marked by an individual color, cluster together in one big cluster. So usually a cluster of DNA methylation, as expected, would define one distinct tumor type. However, we identified 3 clusters in orange, in bright orange, medium orange, and red that combined multiple tumor entities associated with the DICER syndrome and arising at different anatomical locations. So exactly those tumors I talked to you about that share morphological similarities. And on the right-hand side, you can appreciate the same analysis in a 2-dimensional graph where you can again see those 3 clusters, how they tightly cluster together. So these, we believe, are 3 molecular groups of mesenchymal tumors with DICER1 alteration, which I would like to present to you in the following slide. What is the characteristic alterations? How do they look? How do these tumors behave? So to get started, I'll talk to you about low-grade mesenchymal tumor with DICER1 alteration. This methylation clustering group of tumors included tumors from different anatomical regions, so head and neck area, the lungs, the kidney, the uterus. And what we found molecularly was that all of these cancers harbored a DICER1 or multiple DICER1 pathogenic variants, but there was no other alterations in any known tumor suppressor or oncogenes. Looking at the copy number profiles, which are you can find in the lower right corner, these are cumulative copy number alterations of all the 22 tumors we analyzed, and you can see there's not much going on here. There's a little bit of a gain in chromosome 8 and a bit of loss in chromosome 19, but other than that, there's not much going on. And all of these tumors, irrespective of where they developed and where they arose in the body, showed somewhat of a cystic appearance that you can appreciate in the left, whereby very monotonous mesenchymal stroma, which you can appreciate down below, was found to expand below non-neoplastic epithelium. So this morphology was shared across all of these cancers within this methylation cluster. The second cluster we identified is called sarcoma with DICER1 alteration. And in this group also comprised tumors of many anatomical locations, again, including head and neck region, the lungs, the kidney, and the uterus. And in contrast to the previous cluster, in this group, we identified alterations in KRAS, NRAS, and TP53 alongside those DICER1 pathogenic variants. What was very interesting is when we looked at copy number profiles within this group, was that almost 80% of these tumors showed a gain of chromosome 8, alongside other more broader copy number alterations. Morphologically, all of these tumors, irrespective of where they arose in the body, showed a very sarcomatous phenotype, so a solid growth of malignant spindle cells, again with hyperchromatic nuclei, irregular nuclear contours, and where this sarcomatous component was meeting with pre-existing epithelium, again, we saw that kind of cambium layers of condensation of these malignant cells underneath a benign epithelium. And so the last group that I want to talk to you about that we identified is called primary intracranial sarcoma with DICER1 alteration. It's a bit of a complicated name. And this group was very interesting because it almost exclusively was made up of brain tumors, so sarcomas that are found in the brain of young children typically. However, there were also 2 cases of a lung and a kidney tumor found in a specific DNA methylation cluster. Molecularly, these tumors showed, again, DICER1 alterations in KRAS, NRAS, TP53, but also NF1. And based on copy number alteration, we found very broad and irregular chromosomal aberrations. I want to point out that in this group, there was no significant tumor, not many tumors that showed a gain of chromosome 8, as we have seen in the previous group. Morphologically, these tumors consisted purely of sarcoma, so solid growth of sarcoma, no epithelium layer. The sarcoma cells that you can appreciate on the left, with very atypical, very large, very hyperchromatic, atypical nuclear membrane, and we often found high mitotic activity and necrosis throughout the tumors. So does this classification have any clinical relevance? Does it mean anything? And this is what we looked at when we compared outcome data for these 3 groups. And what we found is that indeed, these 3 groups are associated with differences in survival, with a significant difference between progression-free survival of the low-grade tumors and the sarcoma groups, and a significant difference in disease-specific survival for the primary intracranial sarcoma. So it seems that this sarcomatous phenotype in the latter 2 groups is associated with poor outcome in these tumors. So what do we take from all of this? What do we learn for these tumors? What can we use? So what we think this data means is that all of these tumors, irrespective where they develop in the body, likely arise from a shared mesenchymal progenitor, and that by acquiring these additional genomic alterations beside a DICER1 alteration, it confers a progression from a non-malignant tumor towards a highly aggressive sarcoma that is often deadly. Now, our data also points at that there's probably somewhat of a distinct pathway for brain tumors, as we don't see that kind of progression from a benign neoplasm towards a malignant neoplasm. So that is a very interesting observation. Now, however, the problem with this data is that this is only a snapshot of tumors. We're only looking at tumors at a certain point. Can't really use that data to understand how all of these tumors develop. So going forward, it will be very important for the development of new therapies and exactly understanding how these tumors develop, to understand what's going on at tumor initiation. So how is it that this non-malignant mesenchymal progenitor actually leads to the development of a tumor, and what are the exact mechanisms that drive the progression from a benign neoplasm towards these highly aggressive sarcomas. And to study this, of course, you can't only use human samples. Again, that's only a snapshot for most of the part, at least if you look at tumor samples from clinical routine. What you need is model system to study this. And this is why I'm very, very excited and happy that in September of last year, I was able to join David's group in working on genetically engineered mouse models. David and his group and Yemen, as he already pointed out, with many other great scientists, have been able to develop 2 genetically engineered mouse models of these neoplasms. If you're interested in it, there's a one publication. One of these mouse models has already been published last year by Yemen as a first author. And so I was very excited to hear that we were kind of working on a similar topic, and we would be able to join forces to work on this. And so when I first sat down with David in Yemen and had a chance to look at these murine tumors, again, I'm a pathologist, so first thing we do, of course, is look through a microscope. That's just what we love. I was very excited. I was very excited to see that what they had created in their mouse model, to me, looked exactly like what I have seen in human tumors. So this is an example of 2 kidney tumors. One is from a mouse, one is from a human. And I mean, even as a pathologist, I think you would have to take a wild guess which one is from a mouse, which one is a human. Any guesses in the room? Which one is mouse? Which one is human?
I know.
Yeah.
I did like pathology, human curricula. Many similar from the human with the first one.
Very good. I think you could join our research group, no problem. That's exactly the case. The left one is the mouse tumor and the right one is the human tumor. Yeah, I was very surprised to see how similar they were, and I do hope that this will really help us understand these tumors better and lead to development of treatment for these very aggressive tumors. So with that, I want to summarize what I've been talking to you about. First, you've learned about DNA methylation analysis, how we can use this area-based approach and what's behind the principle of using that for tumor classification. I've given you an example of how this methodology can be used or will hopefully be used in clinical practice. And lastly, I've shown you how this methodology has been an incredible tool in informing us on the biology of DICER1-associated neoplasms. And with that, I'm at the end of my presentation. There is, of course, many people I have to thank for supporting me over the last few years, my research, my ideas, some of whom you might recognize on the slide. And of course, I want to thank all the entities that have supported my research, especially the German Research Council that is enabling me doing my research here for 2 years. And then thank you guys for listening, and I'm happy to take any questions.